Hello. Actually, I'm not going to talk about that at all, but it doesn't matter. Vaguely that. What I'm going to talk to you about today is the FP7 funded project KiNDREd that I've been scientific coordinator of since September 2013. KiNDREd is an acronym for kinetoplastid drug development, and we're interested in strengthening the preclinical pipeline. And today, I'm not going to talk to you just about fragment-based design. I'm going to talk to you about our whole approach to drug discovery, because I think it fits quite nicely with what we've been talking about today. So KiNDREd is quite a large consortium. We have 14 partners based on 4 continents, and we have 5 SME partners. Now, the interesting thing about our SMEs, all of them except for us are new to the area of neglected tropical diseases. So we had to persuade them that they really wanted to work in this area. So on the right, you'll see my list of academic partners, all of whom have got pretty good credentials. And on the left are my SMEs. And quickly to see how we are in the world, you see our main focus is in Europe. Oops. Because a lot of us are from France. We have Jim McKerrow's lab in California. We have 5 crews in Brazil. We have India. And we also have someone on the team who comes from Burkina Faso. So, our parasites and our diseases. Our first parasite is Leishmania donovani, but we also work on infantum, which, as we know, causes leishmaniosis. We also are working on Trypanosoma cruzi, which, as we all know, causes Chagas disease. And our final parasite is Trypanosoma brucei for hat. So, we are working on all 3 because that is what the EU call was for. So, our aims and objectives are to strengthen, inform, and advance the current preclinical platform for drug development pipeline, from the early target discovery, validation and screening, through to advanced toxicology and animal testing. And our overall aim is at the end of our 3-year project, we will have advanced at least 1 new lead compound for each of these diseases into phase 1, which sounds very, very ambitious, but you must remember that when we created the project, a lot of our partners already had hits that they'd moved quite significantly along the pathway. So we're not doing it directly from square one, but I'm going to talk to you today about our square one, if you want to say that's our physiological and target-based screens. Okay, so this is our pipeline, very beautifully drawn here. And once our compounds have passed through this phase here, we go into a high throughput in vitro screening platform that's based in an SME in Spain. And the compounds that get through here move through to small animal efficacy and toxicology. But what we've done in this consortium, not only have we got our set criteria for something that is yes or no, we've also put the standard compounds that are used today for each of these illnesses into the pipeline. So we know at each stage whether our hit is as good as, if not better, than what we call our gold standards. So the first libraries we were interested in were compound libraries. When we wrote the project and we thought about the project, compound libraries were what we thought we'd be screening. But, as you've seen, in the time from when the project was conceptualized and actually came to fruition, a lot of other people, like GSK, had done an awful lot of compound screening. So why would we do the same thing? No point. So what we have been looking at are repurposed drugs, and we had access to about 2,000 drugs that are FDA approved. So we've put those through the pipeline against all 3 parasites, and we have got a few interesting hits from those. The only interest in something that's FDA approved is if you're not going to alter it at all. Otherwise, the minute you start changing it, it's no longer FDA approved. So we're only looking at things that really have been used orally, can cross the blood-brain barrier already, that type of drug. But probably I will tell you a little bit about our high-throughput screening very, very quickly because it's a platform. But it's quite a nice platform. We have whole cell screening assays. state-of-the-art image-based screening for intracellular parasites for T. cruzi and Leishmania, and we also have reporter-based assays for 2 cruzi. This is all based in San Diego, so they sent us a nice picture of the beach, and this is all their fancy equipment that they have there. This is all state-of-the-art, it's all brand new, and it's all for us to put compounds through. So, going back to our pipeline, something that we've been talking about today, and I'm going to spend a little bit more time talking about because I think it's something we haven't mentioned too much today. Well, we have mentioned, but we haven't gone into detail, and that is our natural compounds. So, our natural compounds go through our pipeline. Why are we interested in natural products? Well, we said earlier that natural products are a new chemical space. 34% of all approved small molecule drugs over the past 30 years have either been natural products or derived from natural products. So in our consortium, we're very fortunate in having 2 groups, one at the University in Frankfurt and again, University of California, San Diego, who've got access to very interesting natural compounds. Their sources are unusual microbes, and in the US, they're using marine microbes. And I hope no one's scared of spiders, because in Frankfurt, we're using a very interesting source of natural products. A lot of these are entomopathogenic bacteria. These are bacteria that infect insects from 2 species that I've only ever heard pronounced in German, so I'm not even going to bother pronouncing them. Also fungi, plant entophytes, and insect-infecting fungi and rare fungi. And just to show you that natural products are not for the faint-hearted, here is a crab spider that has been infected by fungus, which they've actually isolated very interesting natural products from. That's his fungal infection there. So, the natural products that come from Frankfurt, the crude extracts have been put in the top of the pipeline, so they're going through the whole pipeline as crude extracts. But when we started the project, they also had 120 compounds that they'd already purified. Most of these were from the insect-infecting bacteria, and they've already been tested in vitro by the Swiss Tropical Public Health Laboratory against T. brucei, T. cruzi, and Leishmania, and also against L6 cells for cytotoxicity. So far, we've got 5 active compound classes that have been detected, and there are 5 publications already come out from the consortium just up until February this year on these natural compounds. In the US, they've got a slightly different approach to things. They screened, these all come from, they've been given to the consortium by the Global Health Library of Marine-Derived Natural Products. They have 6,000 selected prefractions that they do a pre-screen on. Now, each of these prefractions contains up to 20 compounds. They've not been purified, they've all been put together. So in total, they're screening 120,000 individual compounds. So these are then put through a non-specific toxicity filter, and anything that's highly cytotoxic is removed from the screen. And when they did this, they got down to just over 3,000 selected prefractions, which they then put through antiparasitic screening, and anything that came up with 80% activity against the parasites was then put through, was considered as a hit, and this came as 102 prefractionation hits. What they do then, this is just the screen against T. cruzi. What they did then, because these are still mixtures, they have to separate them by liquid chromatography, and then they screen what's called a peak library. That's the peak from the chromatogram, and from these they had 33 hits against T. cruzi. So we're investigating those further in the consortium. So the final thing, which was what I was going to talk about, is our rule of 3 libraries. We've selected various targets because everyone who came to the consortium had their favourite target in each of the parasites. So we're looking at these as in structure activity reactions. we've talked about fragments earlier. What we're doing is we've got much, much smaller fragments. And rather than lead compounds, we're looking at much, much smaller molecules. Therefore, we've got, with a smaller number of molecules, you've got more chance of having a hit. But these have lower affinity, but because of the low molecular weight, they have a higher binding efficiency. And over the past 10 to 15 years, they've been considered quite good starting points for new drugs, as Alan Fernlam showed us. In the consortium, we're lucky to have different approaches for fragment-based design. We've got access to SPR, NMR, thermal shift, structure-based fragment design, high concentration bioassays, but also Terry Smith in St Andrews has just done direct wholesale screens with fragments, and he's actually come up with some good hits. This is our SPR platform, which is based in one of our SMEs in Strasbourg. And, we're particularly interested in their fragment here, and they've got up to nearly 25,000 different fragments. These are put onto a chemical array. This is all proprietary based. The compound, the fragments and the chips are all proprietary based. The recombinant protein is then washed over the chip, and it binds, and you see the shift from the SPR readout. And the hits are read out here. You then put that back to your original plate, and you have to remake the product, the hit, once you've identified it, because these all have linkers on to fit them onto the plate. So this screening is really, really efficient because you only have to produce the protein once, you wash it over once, and you get a really nice result. And we've had some very interesting results from this technique that we're now moving forward. Thermal shift works for some proteins, doesn't work for all, which is why, because some of our targets have been very hydrophobic and they just are not very keen to work in thermal shift. We're also doing structure-based design, but for structure-based design you need to have a structure, and for some of our targets we've had to do de novo sequence up de novo structure before we could actually start the study, so those are ongoing. And then in my company, which is also known as Photeomix, we've been doing multiple recombinant targets from the parasites, including the sirtuins, proteases, and kinases. We have high throughput screening against fragment libraries. We've identified hits. We've got IC50s for them. We've put them through the toxicology screens, and we're now moving forward with those as well. When the EU funded KiNDREd, they also funded 3, they have subsequently funded 3, they funded 2 at the same time, other consortia, and the EU are very keen because there's complementarity as well as overlap between these projects that we work together. So we've started working together since nearly 18 months now. KiNDREd and NM-TRIP1 have got an ongoing application for a Marie Curie fellowship. As we saw this morning, the EuroLeash, we have a similar thing, but for all 3 parasites. And with Ray Pierce from A Paradise, who is here, we are working to get a French government-funded PhD student to work between the 2 consortia. So we are synergizing very nicely. So that just leaves me to thank you very much for your attention.